This presentation is in response to exam 3B, question number 1 concerning the positive and negative selection of thymocytes. So thymocytes are immature T cells. All T cells originate as hematopoietic stem cells in the bone marrow. Those give rise to lymphoid progenitor cells, which migrate to the thymus via the circulatory system. Once there, they divide and generate a large population of these immature thymocytes. The earliest of these will express neither CD4 nor CD8, and we classify these as double negative cells. But as they progress through their development, they become double positive cells expressing both CD4 and CD8, and finally mature to single positive, expressing either one or the other. And then from there, they're released into the peripheral tissues. There's some evidence of double positive T cells in the periphery, but their prevalence and function is uncertain. Broadly speaking, the double negative stage is focused on producing a functional beta chain for the T cell receptor. The double positive stage is focused on producing a functional alpha chain for the T cell receptor. As the developing thymocyte moves on through the double negative stages, will express a constant alpha chain, but rearranges the beta chain locus. If the rearranged beta chain successfully pairs with the constant A chain, signals are produced which cease the rearrangement of the beta chain, and this results in cell proliferation. Now, these signals require the pre-T cell receptor at the cell surface, but they're independent of ligand binding to the pre-T cell receptor. So these thymocytes will then express both CD4 and CD8 and progress to the double positive stage, where alpha chain selection takes place. If the rearranged beta chain does not lead to any signaling, the cell will die via neglect. So that's what we call beta selection. Moving forward, positive selection will select for T cells capable of interacting with major histocompatibility complex, MHC. Positive selection will involve the production of a signal by double positive precursors that express either MHC class 1 or 2 restricted receptors. The signal produced results in RAG gene expression, long-term survival, migration into the medulla, and differentiation into mature T cells. So double positive thymocytes move deep into the cortex of the thymus, and there they're presented with self-antigens. The antigens are expressed by cortical epithelial cells on MHC molecules on the surface of those cells. Only those thymocytes that interact with MHC class 1 or 2 appropriately, that is to say, with the appropriate affinity, they can't bind too strongly or too weakly. If they interact appropriately, they will receive the survival signal. Those that do not have the appropriate affinity will die via neglect. And so this whole process ensures that the selected for cells, the selected T cells, will have an appropriate MHC affinity. They must be able to appropriately interact with the MHC in order to affect their immune responses. And so the vast majority of thymocytes end up dying during this process. In fact, a thymocyte's fate is determined during positive selection. Double positive cells that interact well with MHC class II molecules will eventually become CD4 positive cells. Thymocytes that interact well with class I mature into CD8 positive cells. T cell becomes CD4 positive by down-regulating expression of its CD8 receptors. If it doesn't lose the signal, it will continue down-regulating CD8 and become a CD4 positive cell. But if there is signal interruption, then it will stop down-regulating a CD8, switch over to down-regulating CD4, and instead become a CD8 positive cell. Now, this process does not remove thymocytes that may contribute to autoimmunity. So these are removed in negative selection. Negative selection removes thymocytes that are capable of strongly binding with self-MHC peptides. Thymocytes that survive positive selection will migrate to the interface between the cortex and the medulla of the thymus. While in the medulla, they're presented with self-antigen on the MHC complex of epithelial cells. These epithelial cells must be autoimmune regulator protein positive to properly express self-antigens from all tissues of the body on their MHC class I peptides. Some of these endothelial cells are phagocytosed by thymic dendritic cells. This allows for presentations of self-antigens on class II molecules. Positively selected CD4 cells must interact with MHC class II molecules. So antigen-presenting cells which possess class II must be present for CD4 T cell negative selection. Thymocytes that interact too strongly with the self-antigen will receive an apoptotic signal, triggering cell death. However, some of these cells are selected to become Treg cells. The remaining cells exit the thymus as mature, naive T cells. This is essential to central tolerance. It serves to prevent the formation of self-reactive T cells. So beta selection is the first checkpoint. Next, positive selection checks that T cells have successfully rearranged their alpha locus and are capable of recognizing the MHC complexes appropriately. And then negative selection removes T cells that bind too strongly to self-antigens, and together these selection processes establish self-tolerance.